What if I told you that for a fruit fly, deciding whether it's going to be male or female, well, it has pretty much nothing to do with the Y chromosome. Today, we're going to unpack this really intricate genetic program that works just like a beautifully complex set of dominoes, and believe it or not, it all starts with a simple count.
So the big question is, how does a little fruit fly embryo even decide? We're all used to thinking about the simple XX or XY system, like in humans. But for the fruit fly, it's a much more elegant story. It's this incredible molecular decision. And like I said, the Y chromosome is barely involved at all.
Okay, so let's really dive into how this works. First thing, just forget the Y chromosome as the main decider here. For flies, it is all about a cascade of events, this genetic domino rally I mentioned, where one gene's action perfectly, and I mean perfectly, triggers the next, all leading to one final, definitive outcome.
All right, Section 1, the chromosome count. It's all about the ratio. So what pushes that very first domino? The initial signal, the thing that kicks this whole process off, isn't a single gene or a chromosome. Nope, it's something way more fundamental. A simple but incredibly powerful mathematical ratio.
And this simple calculation, this is the crucial starting point. The cell literally counts its X chromosomes and then compares that number to how many sets of autosomes it has, the non-sex chromosomes. This ratio, what we call the X to A ratio, is what determines which path the fly is going to go down.
So let's see how this math actually plays out. Check out this table. If an embryo has 2 X chromosomes and 2 sets of autosomes, well, that gives it a ratio of 1.0, and that is the trigger for the female development pathway. But if an embryo has just 1 X chromosome, its ratio is 0.5, and that starts the process of male development. It's really that clear-cut.
Okay, next section, flipping the master switch. And we're talking about the sex-lethal gene. So the cell has its number, right? But how does that ratio actually flip the first domino? Well, it does it by controlling a single absolutely critical master switch in the fly's genetic code. And folks, this is where the real action begins.
Yeah, this master switch is a gene called sex-lethal, or SXL for short. I want you to think of it as the first and most important domino in the entire line. It is the central control point, and whether it gets switched on or stays off, that one decision dictates absolutely everything that follows.
But this switch isn't just flipped easily. Oh no. What's really happening is a battle of proteins, and it's all dictated by that X to A ratio we talked about. You see, genes on the X chromosomes are busy producing activator proteins, and they're trying to pull that SXL switch on. But at the same time, genes on the autosomes are making inhibitor proteins, which are desperately trying to keep it off. It is a genuine molecular tug of war.
So, here's the outcome. The crucial point is this. When you have 2 X chromosomes, that high concentration of activators just overwhelms the inhibitors. The activators win, and the SXL switch gets flipped on. That is the key to becoming female. But with only 1 X chromosome, the inhibitors win the battle, and the SXL switch stays off.
Section 3, a genetic chain reaction. The cascade begins. Okay, so the master switch is set. That first domino has either fallen or is still standing. And now, a really precise cascade of genetic events is set in motion. The rest of the dominoes are about to follow.
So when that SXL gene is switched on, it produces the SXL protein. And this protein isn't just a bystander, it's an active player. Its main job is to control how the next gene in the chain gets processed, a process called splicing. Think of it like editing a sentence, cutting and pasting words to make sure the final message is correct. SXL's job is to ensure the female pathway continues exactly as it should.
And here is that domino effect in action. This is the female pathway when SXL is on. Step 1, the SXL protein gets produced. Step 2, that SXL protein makes sure the message from the next gene, called transformer, or tray, is spliced perfectly. Step 3, this creates a full functional tray protein. And then, step 4, that tray protein directs the splicing of the next gene, which is called double sex. And finally, step 5, the double sex gene produces the female version of its protein. See, one thing leads to the next.
But what happens if that first switch stays off? What if that first XX domino never falls? Well, the system doesn't just crash. It just proceeds along a different pre-programmed path. This is the default male pathway taking over. So here's what happens in the male pathway when SXL is off. First, no SXL protein gets made. Because of that, the trad gene's message is spliced all wrong, creating a completely non-functional, useless protein. And then, since there's no working trap protein, the double-sex gene just gets spliced into its default form. And guess what? That produces the male version of its protein. The whole chain reaction just goes down a totally different road.
All right, Section 4. 2 scripts, 2 fates. A final outcome. So this brings us to the very end of the line, where these 2 different versions of the DSX protein, well, they act like 2 completely different scripts for development, each one with its own very specific set of instructions.
That's right, the double-sex protein is the final director of this whole show. It's the one that activates female-specific genes while repressing the male ones, or it does the complete opposite. It's the protein that actually builds the fly, shaping all the physical characteristics that make a fly either male or female.
Okay, let's just take a look at the whole cascade at a glance. It really lays it all out. For female development, you start with an X to A ratio of 1.0. That turns SXL on, which makes a functional TRA protein, leading to the female DSX protein, and finally a female fly. For male development, the ratio is 0.5, SXL stays off, you get a non-functional TRA protein, which gives you the male DSX protein, and boom, a male fly. 2 totally distinct routes, 2 different fates, and it all starts with that one simple ratio.
So, at the end of the day, this whole complex system, the molecular tug-of-war, the genetic domino rally, it all just boils down to 2 distinct developmental pathways, both determined by that one initial trigger. It's just a stunning example of biological efficiency, and it's amazing to think it all begins with a simple count.
I mean, when you see a system this complex, this elegant, in a tiny fruit fly, it really makes you wonder, doesn't it? If life's code is this complex for a fly, what other incredible, elegant programs are just hiding in our own DNA, waiting to be discovered? It really leaves you thinking about all the other genetic domino rallies playing out all the time, in all of us, defining every little detail of life.